First up, I should say, we have Dr. James Lyons Weiler. He's visiting us from Pittsburgh, which is very exciting. He holds degrees. He's a fascinating guy. If you get a chance to call him and just ask him he can talk about almost anything, having degrees in biology, zoology, a PhD in evolutionary biology, postdoctoral studies in molecular evolution and functional genomics. His books include Ebola, An Evolving Story, and Cures versus Profits, Successes in Translational Research. Books are available in the back if you haven't seen them. You can there's a sampling of books that you can take a look at. He will also, today he will be presenting results of his review of 2,000 studies on autism that is the subject of his newest book, The Environmental and Genetic Causes of Autism. He will also share with us the results of IPAC's new study on aluminum adjuvants in the pediatric schedule, which are quite revealing and to the point of staggering, I would dare say. Please welcome to the stage, Dr. Lyons Weiler.
Perfect, thank you. So, Leah, fantastic. Where's, thank you. Where's, thank you. That concludes our seminar for today. No, seriously, it's just amazing. I'm like, you saved me so much time in my talk, I can't tell you. Let me just, thank you for the introduction, and thank you so much for having me here. The Washington group for Informed Consent is just an amazing, it's outstanding group. I'm stellar companions, compatriots, in this group, and I am just absolutely thrilled to be here. I'm going to cover a lot of material. Most of what I talk about, a lot of what I'm going to talk about is in the book on autism, but some of it's quite new and currently under peer review. IPAC is pure public charity. Our bylaws state that no one who works for IPAC can have a conflict of interest in terms of personally profiting from the intellectual property or the information that we generate. This is our great advisory board, and the guy in the back there is Dr. Rick Ricketson, who works diligently through bioinformatics and computational biology. He's an MD who's chosen to work with us. You know enough about my background. Thank you for saving me that. These three books were a journey to me. So, first of all, I want to know, raise your hand if you believe that your lives have been impacted by vaccine injury. Raise them up. Okay, thank you. Now, raise your hand if you're in the medical or public health profession, please. Thank you. You're all very welcome to be here. Thank you. And raise your hand if you're law enforcement. I'm just kidding. Sorry about that. So we do have to get along. So you guys would run me out of town if I only wrote the book Ebola and said, there, I've written a definitive work on what I know and think about vaccines, because I talk about how dangerous you all are. What I now call vaccine risk-aware individuals. I use some pejoratives in there that, I really have to edit that book. But I went to write a chapter on vaccines in the second book, and the first part of that chapter on vaccines reads exactly like any news story you'd pick up today in the media about how vaccines have saved millions of lives and so on. And they work and they're safe. And then what about what about Andrew Wakefield? And I stopped. And I'll give myself a little pat on the back because I stopped and I actually looked, right? Well, thank you. I found Brian Hooker, I found the William Thompson story, and so the rest is history. I just chronicled everything that I found in that book. It was just perfect to say, okay, here's the emails, and here's what they said, and so on. And then I sent the manuscript away, and then Vaxxed hit. And I'm like, oh, wow, this is going to be huge. So, but if the CDC is misleading the public on vaccine safety leading to autism, that still is not the scientific route to saying, aha, vaccines cause autism. That's not the science that's used, right? So a magician pulls a fast one on you all the way, and you still don't know where the P is under the right? Shells. So I went and I read these 2,000 studies. I scanned, 3,000. I read them to answer the question that the Institutes of Medicine balked on. They didn't have a free weekend to look at the animal studies and the cell line studies, and they didn't look at the causality in that way. That's literally the interpretation that they gave. We don't have a free weekend. If you go down and look at the IOM minutes that were secured by SafeMind. And so I did that, and it took a while to do it, but I learned so much and it was so worthwhile. I do have some conflicts of interest to disclose. I do not receive income from the manufacture, sale, or distribution of vaccines or any other medical product. I was compensated last year for 2 vaccine injury cases in Ohio. I was paid less than $10,000 total. I do receive payment from donations, and any book that you buy here today or purchase here today is not a purchase, it's a donation. We're not allowed to personally profit and so on. I'm here today because our society is fundamentally broken and it's getting worse. Mish Michaels was a meteorologist. She had a great career. I knew her work because I lived in New England years ago, and she was going to be the scientific correspondent at WGBH. And when they found out that between jobs she testified to a legislator about her concerns over vaccine safety, they dishired her. They decided that she wasn't going to have the job. And encroachments on labor rights are something that we really need to start looking at. I'm sure all of you can think of examples where people's jobs are on the line because they have knowledge about vaccine risk. Autism is certainly on the increase, there's no doubt about it. A percentage of this is, or was, you could say, due to changes in the diagnostic criteria, from DSM-4 to DSM-5 and making autism into a spectrum and so on. However, those changes, maybe, let's give them 30% of that. We still have 30% of an exponent is still a huge increase from 1 in 10,000 to 1 in 68. This is 2004 data, we really don't know this because the CDC on that last estimate said we don't know if it's 68 or 100 or 24 because we do poor estimation. So I looked at autism with a fresh light. I looked at DSM-5, I looked at DSM-4. It was a bit of a slow-moving target. Now there's discussion on that. They want to reclassify autism every day if they can. They want to make it move so far and so fast. And I'll tell you the reason why they want to do that. They want to do that because if you look at every mutation impacting any gene, then you can have a syndrome. And they'll just slice and dice. They'll kill autism by 1 million cuts, and it's not autism. Some of you know that they've done this, and they call it channelopathy, or they'll call it mitochondrial dysfunction. And it's an excuse in court to say, listen, this is a genetic disorder. So I basically regurgitate DSM-5 here for the sake of being accurate, but some of your compatriots in the vaccine risk aware movement got wind that I was working on this book, and talking with them, they said, you need to tell us what happened to regressive autism. We don't hear medical doctors diagnosing regressive autism. We don't hear about that as a medical thing anymore, and yet it used to be studied. You look at the studies now, they don't have regressive autism as a clinical group that's under study, and what happened to it. And where it is, it's in the ifs, ands, and buts here in DSM-5, and I go to some length in the book to do that. There's discussion now after a lengthy law review of all the case histories in 50 states. What's the history of using medical neglect and accusing parents of exacting medical neglect on their children by vaccine refusal? And they didn't find any. They found just a handful of cases. And so this conclusion, the conclusion of this review, of course, was that all the states should now start looking at this as an option because there's no legal precedent for it. That's ridiculous. It's a non sequitur. So as a biologist and an evolutionary geneticist, this is a fascinating disorder to me. It's a fascinating condition. It's very complex. There's 850 genes that have been known to be involved in autism one way or the other, but no gene can say to be liable for less, for more than 1% of the condition. 20% of autistics have higher copy number variations. This is one of the most important scientific findings that I can emphasize today. There is a higher level of genetic diversity in kids with autism, 20% of them, and if we can unravel that, then we can understand what's happening. We have 20% increase in genetic variation. It's impacting genes all over the place. No wonder why there's 850 genes that are associated with it. But remember, if each gene is associated with less than 1%, then if you were just to look at one gene and you looked at the whole population, you would say, that gene's not associated. Now, I want you to remember that from a study design perspective. Regulatory genes are definitely important in autism early in brain development. But the synaptogenesis occurs throughout development and throughout life. I'm doing it right now, by the way. The familial and genetic risk, we have to look at a difference between family risk and genetic risk, right? So family risk is like putting the meatloaf in the oven after cutting the ends off because your mom did it, only to find out that it wasn't because it gets the heat into the meatloaf, it's because grandma did it. That's family risk, right? There's shared environment in the house, and for siblings, there's shared environment in the womb, and those, whether it's environmental exposures or diet or anything like that, these specific risks, these family risks, are not genetic, and yet they can look like genetic to a degree. And they're difficult to tease apart depending on the kind of genetic study you do. Every mode of inheritance of a genetic trait is represented in autism. So you're going to have a gene that says, listen, you've got this mutation, you're going to have autism. It does exist, but it's so those mutations are so rare because they're very lethal. They're sublethal, but they're very deleterious. And I'm going to show you some data, what happens if you get 2 or 3 of those and how their percentages in the population. But there's also many genes that have mutations that are not lethal at all. And, you know, we all have what's called genetic load. We're carrying around sublethals, and we don't even see the expression, right? Because mom had one copy and dad had the other. You know, cancer genes are like this. So you have 2 copies of a cancer gene, one's forking, the other one has a loss of function mutation. You have an increased risk of cancer. You don't have an increased risk of, what's inherited is the risk of cancer through other somatic mutations. Now, this is a representation of identical twin in the red and fraternal twins in the blue. The difference between those distributions, each of those data points on the left are traits that are heritable, that are published heritability, okay? And you can see there's a ton of traits that are heritable in the population, right? And so to look at something as complex as a neurological disorder and say, oh, there seems to be some heritability, we really have to be careful how we tease that apart because it could be the heritability just exists in those traits if they're not disrupted by the environment. Anyway, we're going to look at this from functional groups by age. Early on, there's specific genes that are involved in early stage neurological development. And the regulatory genes, the CHD, CH7, and so on, from neural crest formation and neurogenesis, if you have mutations in those genes, yes, you're going to see structural differences early on, okay? But then we also know that there are many genes involved 0 to 2 years in synaptogenesis, and then we move on to pruning. We make our neurological connections, and then we prune things away. That's a complex process, biological process. From 2 to 4 years, you have pruning and myelination, and so on. And throughout brain development, and then all through life, you have to be able to use your mitochondria, you have to do cellular detox, you have to use your serotonin receptors, you have to use your glutamate, and you have to do your synaptic proteins. So these are all throughout development, and yet when you look at the introduction of vaccines at a young age, you can see that it interferes with, or is done at the same time, I should say, with many of these important processes that we're going to go into some detail on. The genetic study, the focus on early development is a little bit skewed in the scientific literature. I took a very commonly used chart here from this study that shows the changes in these important early regulatory genes over the course of early brain development. And that's kind of what they focus on, and they're kind of hinting at, you know, listen, it's early development, you have structural changes, it's genetic, right? It's not necessarily environment. But then if you look at that on the timing of this other slide, there's all these other processes that are important too. And if you look at, say, sex differences in the brain of genes that are expressed in the brain, there are large differences, and those differences change over time. As children age, okay, as human children age. And so just seeing differences and variation in this tells us that it's a dynamic process. There's no doubt about it. A lot going on there. So this comes, yeah, good. This is a reflection of the literature. This is not my opinion. This is from multiple studies. Those rare synaptic proteins that are broken because you have a loss of function mutation, that you're going to get autism-like features, characteristics, combine it with a rare mitochondrial, that's not necessarily so rare, but rare glutamate receptor genes. You get mutations in a subset of genes, but the overall contribution to risk is really low. So this is a reflection of what you guys know. When they say it's genetic, you kind of like are a little upset, right? Well, we know it's not completely genetic. rare de novo mutations that I just told you about, that 20% of autistics have an increase in these de novo mutations, also rare. Compared to the common variants that we all have, if you have genetic variation in a human population, and it's common in 40, 30, 20% of the population have that genetic variation in one of the genes, that's ancient variation. Ancient, old variation, and you can't get an exponential increase. This is what you guys are saying this all the time. You know, you can't see a genetic disease doesn't have an exponential increase, right? You wake up one day and all of a sudden everyone has this condition. And then there's other genes that are not common, but cumulatively individually they're rare, but cumulatively they make up a large percentage of, and their roles are more like the environmental. I'm sorry. Then anyway, the other category I'll show you are environmental susceptibility genes. Now this is an important study, and it's a little difficult initially to interpret it, but this is these are histograms of males and females, where this is the percentage of subjects that have multiple, a single mutation, 2 mutations, 3 mutations, contributing to autism, and then you have the females here. It's the same story. So you can see that, yeah, a lot of people have, you know, large percentage have 1, small percentage of 2, and then practically none have these others. What these dots are here, and this is described in the book, is an odds ratio probability of having an autism diagnosis. And so if you have 1 mutation, you have a very low, almost 0 probability of having autism diagnosis. If you have 2 mutations, you still have, you know, 2 or 3% probability of an autism diagnosis, and so on. And so it says if you have 10 mutations, you're probably, you know, you're going to have a higher probability, but it's still only 20%. Okay, this is really exciting news because it tells us that it's very difficult to have purely genetic autism. In fact, I think mathematically it's theoretical. I think biologically it probably never happens. So the largest genetic studies are these Hallmeyer and Sandin studies. They didn't measure any environmental factors. Okay, they looked at concordance estimates between twins that are identical and twins that are not identical, and they both of them concluded that autism is about 50% environmental, without even measuring an environmental condition. Okay, so we have a lot of room to talk about environment here. Another study that was done took a different methodology approach, of course, and they found a lower room for environment. So if you take all 3 together, we still have plenty of room to talk about environment. And then on top of it, they don't look at genetic by environment interactions at all, right? So if you say there's 50% genetic, 50% environmental, and you didn't look at environmental factors, you have another term to look at in that what's called a model. It's the G by E term, and unless you measure G by E, you can't attribute a percentage of the liability to environment or genetics by themselves. And this is found time and time again, study after study after study, found plenty of room of variation that's unaccounted for by genetics. Okay, so this is what a pedigree typically looks like in autistic families. The males are squares, females are circles, and those with slashes through it are the deceased. So parents of kids with autism tend not to have autism. They don't have autism in this, right? What this looks like to me is that humanity's dipping its toes in a toxic soup, and the toes are our children. And so this tells us also, this looks like an environmental disorder. It doesn't look like genetics to me. What you should see would be genetics is this one's yellow and that one's yellow. And this one's yellow, so that one's yellow. And up here, maybe this one's yellow, so that one's yellow. You'd see some form of scattering of dominant inheritance. Now, this is going to be very interesting to you. So what we've seen is parents of kids with autism don't have autism. And then the kids that are on the spectrum now that are growing up to be adults, they're starting to have kids. Their kids tend to have, oh, wait a minute. Parents of kids with autism don't have an autism diagnosis, but they tend to have autism-like characteristics, okay? Insufficient for a diagnosis. Now, the kids that are being born of kids that are, kids are on the spectrum, they tend to not have an autism diagnosis, but they tend to have autism characteristics. Now, why is that? That's because something's changed. It's because the ASD spectrum folks are changing their environment, okay? So we have to do good science. We have to be objective in our approach to science, and unless you measure environmental factors, you can't know anything. Here's a mom, and she gave birth to 2 twins, and they both have exposures. And through those exposures, whatever they are, they both got sick. What caused that? There's not enough information here to point to anything. Is it mom's uterine environment? Is it genetics? Is it this exposure? What is it? Is it bad luck, right? It could be anything. The way I look at this is if everyone in the population smoked cigarettes, right, lung cancer would look like a genetic disorder. Okay? Universal exposures and never look at interactions and never do studies. Okay. So you have to do different treatments, like vaccinated, unvaccinated, and then we'll see, okay? Fascinating stuff in the literature out there, those 2,000 studies that I read. There have been people who have found genetic by-environment interactions, although they don't call them that. I call them that because I can recognize them. Organophosphates, if you have a mutation in the PON1 gene, you're more likely to have an ASD diagnosis. If you have an ADRB2 mutation, this specific gene, and you have pregnancy-related stress, you're more likely to have an autism diagnosis. If you have a MET gene mutation, you can't detoxify particular types of pollutants like aluminum that's found in traffic-related particulate matter, you're more likely to have an autism diagnosis. This is beautiful one-to-one correspondence of risk. Okay? Everybody in the room probably knows if you have an MTHFR mutation, you're more susceptible to prenatal vitamins because of folic acid. You should get methylfolate. Great study. And then there's a whole ton of other studies that show specific mutations are associated with specific environmental risk, or mutations in specific genes anyway. And so this is a more complete story than we're told by the media. Every day, my inbox gets a hit from a Google search with autism genetics, and this now shows that autism is genetic. They found 7 genes, and now this shows it's genetic. It's not enough information. MTHFR specifically was found to increase thimerosal susceptibility, and it only makes a great deal of sense. But here's one in OROC you want to look at, because this SCN1A variation itself is associated with vaccine-induced encephalopathy. You can't get more specific than that in terms of this is a genetic by-environment interaction. So I'm hoping that the literature review that I did in the book itself will be a sort of a guide to parents and to doctors when they're wondering, what does this constellation of mutations mean? You go to your genetic counselor, you go to your doctor, and they don't really interpret it for you. If you get exome sequencing, it might be a guide. I can't tell you it's medical, because it's not medical, but it's a guide about what science is saying about the mutations, okay? Then there's this other great complex thing that we have to deal with called phenomimicry. So if you have mutations in these 2 genes, CH7 and CH8, you get a loss of function. But if you take valproic acid, you get the exact same phenotype as those mutations. And it's not because they're causing mutations, it's just because the environmental effect is the same. If you have mutations in your endoplasmic reticulum genes and you can't detoxify your cell appropriately, then, or you have thimerosal inhibition of this protein ERAP1, which Bernadette Pager pointed out to me. If you have thimerosal specifically inhibits ERAP1. Now, what ERAP1 does is it sits who here had high school biology? You remember the rough ER and the smooth ER a little bit? And it's the garbage dump of the cell, okay? That was so boring to me in high school. I'm like, yeah, but tell me about the nucleus. What's going on in the nucleus? I'm more interested in the genes, right? So I'm reading about this endoplasmic reticulum, and who cares what garbage chute the cell has? Literally, that's my feeling at the time. But ERAP1 inhibits proper protein shortening inside the endoplasmic reticulum, okay? It has multiple functions. And if you don't shorten your proteins right, specifically your MHC immunological proteins, these are the adaptive immune system, you're going to have a reduction in immunity caused by thimerosal. Now this study, the study authors speculated that perhaps ARAP1 was so toxic that it could be used as a chemotherapy agent, but they determined that it was too toxic to be used as a chemotherapy agent. And if we reflect on a moment for what that means is if you're going to do a study of ERAP1 as a chemotherapy agent, you have to give informed consent to join that clinical trial, and you have to be told the risks. This is going to improperly shorten your proteins, and it's going to mess with your immune system. And it also is going to cause the endoplasmic reticulum to glom up around the outside coat of the cell nucleus. And so you're basically going to be really making your cells very sick. Anybody want to sign up for that? So anyway, phenomimicry, there's other examples here where you have loss of function mutation and it mimics the exact same effect of an environmental toxin. And so how can we tell the difference? How can anybody in a court of law, on the basis of what tie they're wearing, maybe their hair cut or something, or how convincing they are, or charismatic, can convince the judge that this specific case of autism was not caused by environmental exposure because the child had a specific mutation, when the environmental exposures can cause the exact same phenotype, exact same characteristic? Okay, so that's kind of interesting. In this book, I recognize, because of my experience in molecular biology, that the ASD risk genes that are less than 1 or 2% of autism genes, they're identifiable. You can point your finger at it, and you can say synaptic proteins, TSC1, MCT, and CNT NAP2 was just done in a great study I'll tell you about in a minute. But most of them are environmental susceptibility genes. And that's variation that you're just going to tend to be more sick if you happen to be in a toxic soup. And then another 40% or so is variation. These genes have variation that would have impacted that individual's tendency to be more talkative, tendency to be more social anyway, because there's a huge amount of variation in those traits. And it's heritable variation, but then you put them in environmental exposure, and you end up with a serious, more serious phenotype. Now, this is one of the most exciting studies. It just came out. It's not in the book, but it just came out this week, actually. It's done at Rockefeller University, and what they did was they took mice that have a mutation in the CNTAP2 gene, and they looked at gender, males and females with those mutations, and then they looked at exposure to an environmental toxin, environmental stressor that causes maternal immune activation during pregnancy. And what they found was when you're a male and you have LPS, lipopolysaccharide, artificially injected, and you have this mutation, all of these characteristics that are measures of animal sociology and so on, they're highly significant. They looked at the interaction terms. It was great. I wrote to the author, and I'm like, congratulations, you're doing science. It's fantastic to see this, right? And I was quite sincere. So when we look at the structure of a neuron, we have an astrocyte that is very close proximity to where the two neurons come together, presynaptic neuron, postsynaptic, and this is where nerve transmission takes place. And one of the neurotransmitters is glutamate. Okay, glutamate's a signal from one cell to the other, and the astrocytes. Their job is to suck up the excess glutamate that spills out of the synapses, because you don't want glutamate, because it is a signal of neurotransmission and other things. You don't want it just floating around the cell. So they're like little vacuum cleaners. That's like a little green vacuum cleaner sucking it up, right? So if there's anything in the environment that's going to make your astrocyte sick, and it can't work well, and it can't do its job to clean up glutamate, you're going to have a number of things happen, right? The cells are going to kind of dishabituate to glutamate as a neurotransmitter, because they see it all the time. They're going to say, some of them are going to stay chronically excitatory, and some of them are going to say, well, we got that signal, so we're not going to pay attention anymore. It's kind of complex, but when you look at the role of the astrocytes, they're a very important cell, because mopping up glutamate then cleans up the brain, and it says to microglial cells that there's not an infection, there's not a brain injury. So microglial cells are another type of cell. They're the sentinel cells. They're hanging around. They're waiting for an infection. They actually work together. They will hold, hang on, I need two hands to do my synapse modeling. They'll hold hands of two neurons, and they'll say, you two, get together and be friends, and do your nerve transmission. When they're not in their resting state. I think I turned this off. I'm not sure. I'll say. So you can still hear me okay in it? Okay, good. So the microglia, when they're in their resting state, they're not doing anything. But when they're in active state, they're either kind of helping the brain learn complex connections, making neurological, redundant neurological connections at the multiple synapses. And that's how we learn things. It's through reinforcement learning. Like, who here knows my last name? Raise your hand. Quick. Okay. My last name is Lyonsweiler. Raise your hand if you know my last name. Lyonsweiler. Okay. It's going to be tested in 5 minutes because you either have that structure in your brain now permanently, and my book is on Amazon, thank you very much, but you either have that permanently or you don't, and there's a physical representation of memory in that way. The microglia, if they're not available because they are sick by some way and they can't do the hand holding, they can't help make the one-to-one connections, okay? And so the autistic brain structurally looks like many one-to-one connections. And this was through reading so many of the studies. Many one-to-one connections in the brain as opposed to many to many connections in the brain, okay? And when you have many to many connections in the brain, a signal comes from one cell to the next through this synapse, and then inhibitory signal goes back and says, got the message. So how many of you right now are thinking of the fact that you're sitting on your chair? You have forgotten that. You have habituated to that, okay? You're not thinking about the pressure of the glasses on the nose, right? That kind of thing. There's certain signals that the human brain has evolved to always pay attention to. We don't dishabituate normally to things like faces. It's very difficult to not know you're looking at somebody in the face, right? If you've ever noticed that. But you can kind of zone out other things. You can filter out other things. When you have many to many connections, your brain's constantly sending the signal, but it says, no, stop it. I already heard that. That's good. We're good. But if you have many one-to-one connections, a really interesting thing happens. The pressure of the chair is constant. The wind blowing by, you constantly feel it. The light coming in, constantly feel it. The sound of that door, I'm going to hear that for the next 10 minutes. Okay? And so it all becomes overwhelming, and we know this full well. And the other interesting thing that looks to be happening, and this is a hypothesis of mine, is that signal comes in so far and so fast without inhibition from the eyes and the ears and the other senses that it goes all the way to the motor neural cortex. And the motor neural cortex can only interpret nerve signals as an impulse to move. And so autistic kids, because the fan is spinning or because the television is loud, okay, and it's an impulse, they can't handle it, they can't control it. It's not about control. Their brain's working perfectly in responding to the signal. We just don't have the inhibition there. So the microglia are really essential to not be overactivated. And when they become overactivated in the presence of excess glutamate that you can't shut down because mercury or aluminum are in the microglia, and that there's studies that show that, then you're going to get chronic microglial activation. And with chronic microglial activation, then you're going to see these symptoms that we call autism. And the microglia, actually, when they become chronically activated, normally they're supposed to destroy bacteria or do nerve pruning. They're programmed to destroy dendrites for normal neural pruning. But if they're overactivated, they're going to go all over the place, and they're destroying dendrites all over the place. That's why we have the many-to-many, one-to-one, sorry. And then they also destroy neural precursor cells. So the good news is when people have a stroke, Russell Blaylock said this many, many years ago. When people have a stroke, the first thing they do is they try to get the glutamate out of the blood. Why do they do that? Because it spills from the brain over the blood, and they have much better clinical outcomes. Where's the research on getting the glutamate out of the blood during a vaccine injury? We need science, folks. Okay? If somebody has a traumatic brain injury, it's the same thing. They've learned that there's a depletion of glutamate. There's multiple ways to shut down microglia as well. Luteolin is a compound that is known clinically to shut down microglia. I have a list of things in that book that will shut down microglial activation. Will it take the toxins out of the brain that are causing it? No. But will it give kids a better day? I would hope so. So we need science that addresses that autism is an environmental condition and absolutely needs to be looked at that way. We don't need any more propaganda, please. There's plenty of genes that have specific one-to-one correspondence contribution. It's not an algorithm, it's not perfect, but there are many genes that are known to contribute to specific symptoms that are associated with autism. And that's okay, because there's variation in all things in human beings. We have a lot of genetic variation. But there's also a ton of environmental factors, excuse me, that are known and suspected, and none of these will look new to most of you. The studies that show that these specific environmental toxins might be liable exist, and yet you're not told about these. So the book has those, they're reviewed. as well, like acetaminophen. And you guys taught me about that. I never heard that acetaminophen was a problem. Your community taught me that. That was Autism Moms. And I looked at the literature, and sure enough, there's, in 2008 and 2016, 2 different studies, okay? That's a confirmation study. There's autism spectrum, symptoms are more likely, okay? Then, so my conclusion about autism is that I thought I fixed this. I must have the wrong slides here, but it's at least, it's no more than 50% genetic, but it's at least 50% environmental. Now, this is something I want to talk about here. This is my environmental toxin sampling liability concept. The circles represent families, and the families all have unique susceptibility to specific toxins. So valproic acid, organophosphates, particulate air pollution, and so on. And everyone's immersed in the same toxic soup, right? But there's specific risk to specific families. Wouldn't it be nice if we could do a genetic screen and say, hey, you know what? We have, you want to avoid those kinds of toxins in the environment, okay? What if the toxin is mercury, which stays in the brain longer than methylmercury? They had that backwards for years. The aluminum story is as follows. 850 micrograms is the amount that you're supposed to get in a vaccine if you're an adult my age and my size. In a vaccine, that's the limit. And yet, at some of these baby visits, you end up with more than that in a child. And so we got really curious about what this looks like. If you're looking at multiple doses of aluminum over time, and it's known to be a neurotoxin, then if you put that in the context of known to be a neurotoxin, okay, if you put that in the context of this environmental toxin sampling liability idea, and then you add the doses of aluminum for the vaccine, these things can very easily be seen to be pushing kids over the edge, okay? And then aluminum is known to cause stresses of the endoplasmic reticulum as well, just like thimerosal, okay? So it has many toxicity measures. I don't have time to go over this kind of thing. This is the slide that everybody likes, that it shows that, yes, some studies show association with vaccines and autism, but we're moving beyond that now. They haven't tested every vaccine for association with autism, so how do they say vaccines do not cause autism? That's magic, right? So the question is, with the limits being, if you're going to get a dose of aluminum in a parenteral source, that's an IV drip, it is known to cause neurotoxicity and serious problems with the brain at 4 to 5 micrograms per kilogram per day. Okay, that's known. 4 to 5, and remember that, 5 micrograms, right? And that's from the CFR and the FDA. But if you put it in aluminum in a vaccine, and you have an 850 micrograms aluminum per dose, what does that look like with body weight? So we worked that out. We looked at, I don't have time to go into that, but at different doses of, these are milligrams of aluminum per kilogram per day for safety studies. The safety studies that were done looked at dietary aluminum. It wasn't injected, and most of them were done in adult mice and rats. They weren't done in children. They weren't done in baby mice with developing brains. And so we also found an error. The World Health Organization said they had a limit of aluminum of 1 milligram per kilogram per day as a provisional tolerable intake of aluminum, and that was transmitted in this study by another committee of 1 milligram per kilogram. I'm sorry, it was 1 milligram per kilogram per week, got translated mistakenly into 1 milligram per kilogram per day. And that's a big mistake. And so when you look at the dosing and you look at body weight, the black line being a safety level, pediatric dosing safety level that we determined, you can see that the babies are being overdosed for body weight much more. You know, babies are not, little adults, whereas the adults can seemingly tolerate more aluminum. And that is true for specific vaccines as well as for the combined vaccines visits. This is our calculation of the accumulation of aluminum over the childhood schedule, and you can see that it's in excess of a safety limit that we determined when you actually bother to calculate that 850 micrograms that I'm supposed to get, and then correct for body weight, okay? And so we're in excess. Now, Paul Offit would like you to think that you get much more aluminum from diet than you ever would from the vaccines. And he's correct. You get more. You eat more. That's fine. but only 0.1 to 0.2% of the aluminum that you eat makes it into the body per se. It makes it past that epithelial layer, okay? 100% of the aluminum that you inject makes it into the body. And so when you're looking at it from dietary versus injected, the vaccines are actually responsible for 96% of the aluminum in a child's exposure. 93% for 6 to 11 months. A 2,676% increase in aluminum exposure in a newborn. Okay? And so when we look at diet across humans for adults and so on, and then we look at exposures, you can see delay in maturation rates at 108 micrograms per kilogram of rat pups, and for preterm babies, they get 125. You know, we're overdosing our kids with aluminum, there's no doubt about it. And aluminum is known to now 10 micrograms per gram per tissue in people who died from Alzheimer's. Chris Exley found that result. Okay, that's a lot of aluminum, and you can actually see the aluminum in the brain, and it's co-localized with the senile plaques. And so yet the media would tell you that everything's fine, everything's safe, vaccines are safe, and there's no risk. They're effective. They want to change the discussion from safety and risk to efficacy. And yet it seems like they have everything under control, and yet do they really? We can do better. We can take the environmental toxin sampling liability concept and we can say, this is why, you know, kids that have higher amounts of organic pollutants in their serum tend to have worse behavioral measures on autistic behavioral measures if they have autism, but they don't if they're in a control group. This is a group in Pittsburgh that does this. And so these are studies that are coming out now that are finding early changes in the brain, and they're trying to say it's genetic and so on, just because there's early changes in the brain. But this shows that over the course of child's development, up to 48 months and beyond, the brain is a dynamic process. Yes, these are cortical neuron layers that are the last layer of the brain that evolved, in the neocortex. The laminar layers actually go through differentiation and specialization, and the structures may be different on the early onset. But these studies were never looked at vaccines. They never took the kids that had early indicators of autism, right? And then said, well, what about the ones that we vaccinate versus the ones that we don't vaccinate? And yet these are being wrongly interpreted by scientists as saying these actually do not, these allow us to rule out vaccines, and they don't. That's microglial activation. You guys know all this. You do, believe me. That stuff you know. Oh, thank you, Garth. You always heard about the rabbits. So every time somebody posts on social media that the crazy vaccine risk people, anti-vaxxers, should go vaccinate their children, they don't know that it's mostly vaccinated people. I just post a picture of rabbits now. But the AP would explain to you that, you know, hey, the reason why these people are, not every vaccine is perfect. Well, that's new. Okay, let's talk a little bit. How much time do I have, Bernadette? 10. Okay, thank you. So let's talk about rights in America. 15. Yeah, good. Let's talk about rights in America, okay? We have a number of encroachments on our civil liberties and our rights, and I'm not breaking any news here. But, you know, we have something called the First Amendment. Bernadette alerted me to open comment period on a proposed weakening of the MMR vaccine information sheets, the ones they don't give out to patients anyway. That they wanted to weaken them because they thought that they're too scary as they're written. And so there's a call for public comments, and we alerted other people about it as well. We submitted our comment, and we watched as 375 other comments were published on the website, and ours didn't show, and ours didn't show. It was like 12:53 in the morning, and Bernadette or I are online together, and she goes, it's there. And so we looked at it, and this is what they did to our public comment. Bernadette and I had so much important things to say to you that they wanted to interfere with our ability to talk to you in a public forum, okay? And so there is also specifics about aluminum neurotoxicity in there. They don't want you to know about. But then Dorit Reese had submitted a public comment where she said, no, they really do need to be weakened, and here's why. And this is not her comment. This is our comment in rebuttal to her comment. So here's an American citizen trying to talk to other American citizens, and then we talk back to her, and the government says, you can't say that. And what do you think it was that was redacted was the fact that she has conflicts of interest with pharmaceutical companies. So a further redaction was that Dr. William Thompson of the CDC, you know that story, right? So, and we also complained, it's not old news, don't get me wrong, buy Vaxxed, give it to your doctor, you know it. Where's my t-shirt? I love you, Del. Anyway, but they do know it, and that's a win. The fact that you know this story is a major win. Kudos to you guys. But one of the things that she was saying was scientifically incorrect. She did a misinterpretation of science. And so we called her out on that. I want to talk a little bit about informed consent. So you're going to hear a lot about informed consent from other speakers today. But for 16 years, I was trained and tested on informed consent and participation in a clinical trial at the University of Pittsburgh. Excuse me. You have to be told the risks and benefits of a clinical trial. And there's a huge amount of regulatory control over this. And that regulatory control means that people go to jail if they violate informed consent. You know, you can't treat people for things like for cancer with something that, but without special protections. The pregnant women and fetuses are afforded special protections, and children are afforded special protections. I mean, it's very difficult to do clinical studies on pharmaceutical products for children. And then there's something called the common rule that you should learn about that disallows coercion altogether. If you're in a clinical trial and they say, no, you really should be in the treatment arm because if you don't, I'm going to go crash the windows in your car and steal your radio, all right? That kind of thing is ridiculously wrong, right? And yet, you also can, in the common rule, no investigator, the sponsors, institutes, or its agents can be protected from negligence if something goes wrong. So you can't just have the patient consent and then put in the consent, well, if something happens to me in this clinical trial, I'm not going to sue you. And so these encroachments are increasing in diversity and number, and so we need all hands on deck. We need people, you guys need to specialize. You need to have a committee for each of these, actually. You need to go and look at specific ways that people are doing things and then share the load. But CDC and AAP teaches medical doctors that they should manipulate your perception of risk, make people feel like there's a lot of disease out there, make people feel like a lot of people are suffering from the disease. This is what they're actually teaching each other. And then on top of it, we have the medical doctors saying, AAP saying, you know what, kick them out of your practice and deny access to medical care. 47 states have, non-medical exemption clauses, thank goodness, but as you know, they're completely under attack. And it's inconceivable to me that coercion and revocation of First Amendments happened in 2016 and 2017. And in our comment to the CDC, we wrote both persuasion and coercion. No, we didn't write that, sorry. This study, Callgrove actually wrote that both persuasion and coercion are necessary and neither is sufficient. They're actually calling for, in intellectual journals like the New England Journal of Medicine, for coercion, overtly, right out there for everybody to see. And it's illegal. Coercion in medicine is illegal. There's Mary Holland. Everybody give a round of applause to Mary. She stood up to someone who said that maybe political candidates shouldn't talk about vaccine risk. And because it's like yelling gunfire in a crowded theater. And she said, well, you probably should say something if someone's shooting a gun in a crowded theater. And she won that debate, okay? So here we have a situation where they're manipulating the perception of risk while doing nothing to minimize the risk. In fact, they're making it worse. They want to add more vaccines to the schedule. They want to do it to earlier with babies and so on. So that's exactly like telling a child to go play in traffic and telling them, go ahead, there's no cars there. It's exactly like telling this kid that this is a great time. Indoors, okay? And it is. It's a lot of fun, trust me. But 2 days ago, there's a product here, a table saw on a wheelbarrow that was recalled. It's a Sears, it's not a wheelbarrow. These things were collapsing and causing personal injury. And some guy lost a thumbnail, and a couple people got their hands cut or something. And that product is recalled because 11 people got hurt. And so if your risk perception and risk analysis is broken, and then you're going to manage your risks just by ignoring it, someone's going to get hurt. We know there's tons of side effects, there's tons of adverse events, and we're told that it's rare, but it's rare to the tune of $3.4 billion, and each one of these might be relatively rare, but you add them all up, and everybody's getting, not everybody, but a lot of people are suffering here. And so to compare the table saw and a wheelbarrow idea of 11 people getting hurt, and we have over thousands of deaths and hospitalizations here, something is fundamentally wrong with the way that we're looking at risk here. And that's why when somebody calls me an anti-vax person, I'm not, by the way, I'm pro-immunization. I want to make vaccines safer. We can still be friends, I hope, because I want to protect babies the same way that you do. I just happen to have enough technical knowledge, and I'm not pulling a Trump card. It's not about authority. Trust me on this. Okay, I just said trust me on that. I'm going home. But anyway, the point is, if we go for the bait of a anti-vax versus pro-vax debate, we're going to not make any success in actually talking about vaccine risk. They've polarized it. They dichotomize it as a false dichotomy. It happens in marriages. It happens in arguments in couples. You always do this. No, you never do that. I'm the one that always takes out the garbage. Well, I can remember. Okay, so this is juvenile, right? We have anti-vax people and refuseniks over here who have the right under the law to say, no, I'm not going to take a vaccine. And we have people over here like the marketing department and the CDC and trolls on the internet saying vaccines are perfectly safe. But there's a huge spectrum here. And so if you change the discussion from anti-vax to vaccine risk awareness, it more properly lets you advertise that there's risk every time that you do it. Now, here's my media guide, okay, to vaccine risk awareness. An anti-vax person says, I want to ban all vaccines, and vaccines can never be made safe, okay? A vaccine risk awareness person simply says vaccine risk is real, and it should be minimized. Guess what? This is a greater set, because this set includes these guys. They also want it to be minimized, okay? There may be differences in terms of whether they could be made safer, but we'll talk about that through science. And then Toni Bark, this is one of the greatest expressions that I love that she said. If you believe those who are injured by vaccines are anti-vaccine, do you also believe that those who are injured by faulty brakes are anti-brake? The logic of calling somebody anti-vax because they want to save other people's kids is completely warped. And yet they want to hold parents accountable for medical neglect. There's a duty to warn about risk. That's medical neglect. Not giving a person vaccine information sheet, medical neglect. Not telling them, oh yes, you know what, you could get encephalopathy from this. And instead what they want to do is they want to change the topic every time you want to talk about risk. They want to change it to safety. I'm sorry, they want to change it to efficacy. Hey doc, what are the risks of these vaccines? Well, they're very safe, okay? And they work, okay? Well, could you tell me any specific risk? Well, they're just very safe and they work, okay? This is what they're taught to do. I was on the phone with a medical doctor for my child, and conversation went on, and I said, okay, but I really need to know, what are the specific risks? She goes, look, Bob, I'm required by law to tell you that they're safe. And I said, you just said you're required by law. Could you tell me which law in the state of Pennsylvania or the United States government compels you to say that? She goes, I never said that. This is not new, right? Bernadine Healy said there may be genetic susceptible subgroups. Cheryl Atkinson has, had interviewed her. Cheryl Atkinson's another person that we got to talk to, right, about this labor rights. So here's kind of a beginning, a first pass at what would I do for, ethical vaccinomics or safer, personalized immunomics would be. You have to screen the vaccines for parts of the vaccines and the proteins that match human proteins. IPAC has looked at pathogens and extensively. I'm not showing you the data, but there's extensive matches between pathogen proteins and human proteins in vaccines. You put those proteins in the body in the presence of aluminum adjuvant, you're going to get an autoimmune disease. It's very straightforward. And it can find cross-reactive antibodies. And so one of the things I encourage parents to do is when they're dealing with lawyers and vaccine injury is to say, is there a possibility that there's an autoimmune disorder here? Let's take a look at the proteins in the vaccine and look at the serum sample from your child and see if we can identify cross-reactive antibodies and say, look, you put that in my child and now they're allergic to their own protein. If you use more antigen and no or less aluminum and mercury, that's safer. You're just moving the dial towards safer. But the fact that they have some 200-something more vaccines that they want to add to it, that's just a short-term solution. So you need to say no aluminum and no mercury. If you deliver the antigen to the proper tissue, if you inject it in the muscle or inject it into the body past the epithelial layer, you never get a full dendritic response. Thank you. And that dendritic response is critical for long-term immunity. Okay, so imagine you've got just safe epitopes, no preservatives, no adjuvants, and you just put a Band-Aid on your skin or something and walk around for a week or something, and then it peels away, and you end up with immunity. We need randomized clinical trials, not correlation studies. Correlation studies are the worst type of science that you can do in this situation because they're so easily manipulated. We need to compare schedules. It's just science. Bob Sears has ideas. Paul Thomas has ideas. They're outlined in a book back there. There should be a clamoring for clinical trials to say, okay, does delaying or spreading them out reduce vaccine injury, reduce the rates of adverse events of any kind? They only want to talk about serious adverse events. What about the moderate ones? Having a seizure is not normal. And doing nothing medically about a seizure is medical malpractice. It's medical negligence. Knowing that there are techniques of intervention that you should look at, blood biomarkers for seizure to see if there's ongoing brain damage. How about a CT scan and see what's really going on? But to send the kid home and say, no, just bear it out is draconian and it's cruel. And we need to get rid of errors. We need to actively track and report vaccine injuries. And so here's a question I want you to ponder. There's probably not an answer today. You're going to agree probably as a knee-jerk reaction. But this needs deep thought, and it needs a lot of work. If there is a genetically susceptible group that has increased risk to vaccines or their components, and if they can be identified, those people under the Constitution are entitled to equal protection. If you can identify a minority that's at special risk, and the majority doesn't take the step, our founding fathers were so wise, doesn't take the step to say, wait a minute, this person or these people are at special harm through no fault of their own, other than the fact that they were born with an MTHFR mutation. This is a civil rights movement that cuts across culture, it cuts across race, it cuts across religion. This is an American movement to say we need special protection because somebody's causing 20% more mutations in our population. We need to find out, is it pelvic CT scans like Ted Fogarty hypothesized? And so I want to leave you with the thought that your answer to the doctor should be like this. The doctor gets mad at you or something. Can't you do any better? Can't you do better? We think we could, people respond to that, especially males. I respond to it very well. Grace's mom pulled that on me last week, and I was like, oh, yeah, sure, what can I do for you, We can do better as a society. And, I want to leave you with the knowledge that we have 1,000 references that are cited in my book on autism online. It's a great researchable reference. But we have increases in many other diseases other than autism as well. So, knowing full well that things are increasing over time and vaccine are increasing over time gives us a hypothesis to test. And the best thing about my position at IPAC is, so far as I can tell, they can't Wakefield me. Thank you very much. So let me say a little bit about your question. So no, I agree. What I just showed was that the absorption of aluminum in the gut is really tiny, but that's not true if you have lesions in your gut. And there's aluminum in the baby formula. So okay. Yeah. Thank you.
That the homeopathic vaccines have safety and effectiveness, and in the bigger picture, I distrust all of the CDC-approved vaccines and would advise nobody to take them. But are there Is the concept of vaccination itself a valid concept?
Can you induce immunity from artificially injecting or introducing antigens into the human body? The answer is 100% absolutely yes. It can be done. Every study on HPV that has ever been done that looked at the HPV targeted viruses, the types that are in the HPV vaccine, has shown, independent study or not, that the viruses that are targeted by the vaccine are cleared from the population. The problem is, it fails at the population level because there's 104 other types of HPV that come in. They're rarer and potentially more dangerous, and they replace the ones. This is not controversial. Type replacement happens all the time in flu. Everybody knows there's another strain of flu because they change the vaccine every year. With HPV, these other rarer types may be more aggressive, and we could end up by reducing these higher frequency HPV types with vaccination with increased rates of cancer that are related to HPV because the rarer types are more lethal. So, I mean, it's very difficult to say an antigen because it's on the surface of a virus versus an antigen that happens to be in the body is no longer on the virus, is no longer immunogenic. It's just impossible to do that. The literature is filled with thousands and tens of thousands of mice who have died due to the disease because they were given the disease but not a vaccine. Okay? But we don't look at adverse events in mice that way. So the answer is yes.
Can vaccines be taken orally?
Yes, there are oral versions of vaccines. Absolutely.
The other issue with, even though, yes, obviously, vaccines have some effect.
Thank you for your question.
Is non-responders. Oh, excuse me. Non-responders, and I think maybe they'll talk about that later, hopefully. Yes, non-responders
there's gonna be more later.
is a very significant portion of people who get vaccines that just don't work. And
What
we need
broke
to
their immune
identify
system?
those people.
Why doesn't their immune system work? Maybe because they're vaccine injured. Think about it.
Could be.
Maybe because thimerosal messed with their ERAP1 and they can't do their immune system anymore.
And then the long-term effect.
And then they revaccinate.
Yes, booster.
Because they can't find
Yeah,
titers.
Exactly.
I had a
exactly.
quick question when you'd mentioned the aluminum and the mercury. I understand that they're necessary in the vaccines as stabilizers.
Yeah.
I'm just curious, is there another stabilizer that is better that you're aware of?
Well, the one of the ones that they're looking at is lipopolysaccharide as an adjuvant, but I just showed you that it causes autism in mice. So is it better? No. I mean, that's a scientific question. That begs the question, we need to do science on that. If the goal is to activate the immune system, why not give a child access to antigens while they have a common cold? While they're already sick. Hey, you know, take this home, and the next time your kid gets a fever, give it to them. Why not do something really incredibly simple like that? Well, it's, you know, I don't know why. Yes.
Do you know if the common rule you cited with regard to clinical trials is being updated in light of the 21st Century Cures Act, which passed last year just before Christmas?
Is that two
overwhelming-
questions?
No, I'm just a statement.
Okay.
The 21st Century
Yeah.
Cures Act now allows for clinical trials without informed consent, provided that there are, quote, adequate safeguards and the risks are minimal.
Right. So what he's talking about is this clause that was slipped into the 21st Century Cures Act. I just showed you all the regulations that already exist on informed consent. Those have not been undone by 21st Century Cures Act. They have not been done because if you're doing a clinical trial on something like a vaccine to determine if it's safe, how do you know if it's safe until the end of the clinical trial? It's impossible. Yes.
Just a couple of questions. Blood-brain barrier, the immunization in Japan study, I don't know if that's antiquated now, if they're still using that as far as delaying immunity boosters and shots. And then also I forgot my third one. But anyways, the excitotoxins too. And so the whole process of why not wait until at least the blood-brain barrier seals up before they introduce these?
These are all valid questions that could be addressed by science, absolutely. I think a prioritized list of questions would be in order so that we, minimize the risk altogether. We end up with, we're 7 times over what the World Health Organization determined to be the safety level. Using animal studies that were dietary on adult animals, so that doesn't even really count, right? So it's much worse than we could have imagined. It is a hook, it's in our paper, and we out them on that. But What I wanted to say about it is, because I can't really find the slide, what I want to say about it is none of the levels of aluminum that are used in vaccines are determined by safety studies at all. They're determined specifically when we did the provenance of the analysis of how we determined what level to use. It was determined to use 850 because it is the least amount that's known to induce an immunogenic reaction. So they changed the study from safety to efficacy. That's the entire problem here. And then on the well-child baby visit, I don't think we need to compare schedules, actually. I think the fact that we're overburdening the children with safe levels of aluminum means that we really should space them out. It's not a question of can we. I think we actually should. Thank you.
From your opinion, how effective is using methylation in diagnosing kids with being potentially susceptible to ASD?
So you take a methylation assay. Are you talking about a test?
Yeah.
And you're studying the genome to see what parts are methylated?
Deficiencies in
sure what you
methylation processes of kids. I've heard.
Oh, okay,
there's some-
methylation deficiencies. So the HK3ME, if that's functioning or not. Well, here's a kid that has an unresolved immunological problem, an unresolved neurological problem already because he's not methylating. Every process in the brain, gene regulation, I can't believe that you can't get a medical waiver in every state on the basis of autism diagnosis or being on ASD because it is an unresolved neurological condition. And it just seems so, whether it's you're talking about a root cause, waivers and so on, and medical exemptions are given on the basis of phenotype, not biomarkers. You're bringing up the point of my talk, which is we know so many genes are involved in autism. Why are we not screening kids for risk? And thank you for bringing up the question. It's one of the slides that I guess I skipped over very fast. We have 850 genes. Put them on a panel and do a study and say if we screen the kids before vaccination for those that are going to vaccinate and screen kids that are not and say, okay, you're not going to get a vaccine because these genes are indicated as you shouldn't get a vaccine. Can we bring the serious adverse event rate to 0 like next year? That'd be great. I see a question here, and this will be a last one. I got to move on to the next
Okay,
speaker.
final question.
First of all, it depends on the vaccine. They should never say just keep vaccinating because they had an adverse reaction. The vaccine inserts specifically state if you had an allergic reaction to a vaccine, then you need to avoid this or that vaccine. It depends on the type. Second, MTHFR mutations are very common in the population. And that means in a root cause analysis, the factor is not genetic, it's the vaccine. If you didn't get the vaccine, the MTHFR mutation has no particular effect. All right, it might be combined with folic acid. What I would say to doctors who look at a mutation in cancer, it took decades for us to figure out how to look at a loss of function mutation that confers risk for colon cancer. Because each mutation has a small percentage in the population, none of them achieve the level of a clinical mutation, right? So they look at it and say, well, I don't know if this is a medically important mutation or not. But if it's a loss of function mutation, we need to redo how we think about clinical genetics. This is a massive revolution in health. If we do it for this condition and they allow loss of function mutation and medical interpretation across the board, we're going to be helping every other condition. So that's one of the things. You guys are thought leaders, and I love you. Thank you.